3 out of the 5 main ones all target iron or heme, and that's sort of how I became more interested in how drugs work and about mechanisms of resistance by itself. But it also started a long time ago. I grew up about a mile from this statue in Birmingham, Alabama. It was on a steep hillside. If I was able to ride my bike up the hillside, the guard would let me in for free, and I could go take a look at Birmingham. So I think Birmingham did not exist before the Civil War, had all the makings for steel, and so I guess iron was sort of on the back of my mind growing up, tabled for many years. My 2 sons, Ronan and Ben, can tell me about the chemistry of iron, that it's able to conduct electricity and heat. It's 10th most abundant element in the whole universe, although the Earth has, it's the most abundant in Earth with 90% of iron being in the core. On the crust where we live, it's only about 5% by weight, 2% by total moles. Most of it is not accessible in hematite ores, but free iron, ferrous iron, is a necessary trace element used by all living organisms, except for, as Valcolata has demonstrated, that Borriello uses manganese rather than iron, but most all other organisms require iron. It's a double-edged sword in biology. It's beneficial, used for enzymes, hemoglobin and oxygen binding, required for energy production, required for DNA synthesis. But at the same time, it's toxic. It can generate free radicals, can inhibit enzymes as when it's in a free state, and it's actually used by white blood cells to kill bacteria and cancer cells. The chemistry of that killing is Fenton chemistry or Haber-Weiss chemistry that generates the O2 hydroxyl radical that ends up damaging proteins and peroxidizes lipids. And so this is sort of the toxic side of iron by itself. So what in the world is the malaria parasite doing inhabiting a red blood cell with 20 millimolar heme iron and lots of oxygen? So it just goes right to the thick of the battle with cleaves hemoglobin. Oxygen is released, iron, heme is released, and so that could just generate, if you were a white blood cell, this would be just what you want to do, but it's got to sort of survive in that environment. So it does take it, here up on the, on your upper right is transmission EM of the malaria parasite, which has gobbled up about 80% of the hemoglobin. There's the lysosome-like with the dark black area being the hemozoin, and which makes a, so the parasite has ended up making a crystal inside of itself. So crystallographers have it easy. They have pure proteins, and they slowly come up with 1,000 different ways to get rid of water so they can make a crystal out of something. But the malaria parasite has to make crystals inside of itself with lots of proteins, lots of other things around. And that was part of the problem or the puzzle that I was working on 20 years ago. Parasite also lacks heme oxygenase, which our cells use to degrade heme, and although it's out of the pot and into the fire because you got to deal with free iron. And it's also known that when it lives in this red blood cell, that iron chelators effectively kill the parasite, where they're really just cytostatic for bacteria and cancer cells. This is just a picture of an early ring form progressing to a hemozoin containing or heme crystal containing, a scanning EM picture of P. falciparum heme crystals, and then an atomic view of the head-to-tail dimers of the crystals all by himself. We sort of thought about other ways to diagnose malaria, and actually Ronald Ross saw malaria crystals outside the stomach of the mosquito and said, well, this is what's seen in the blood. The mosquito is then implicated. So some interesting properties about this heme crystal is that it polarizes light, it's paramagnetic, and it can absorb laser energy. So with colleagues at the Applied Physics Laboratory, we sort of demonstrated that you can just, without adding any matrix, you can just hit it with a laser and a mass spec machine, and hemozone will give a characteristic signature 5-peak pattern where heme and hemoglobin does not, because it doesn't absorb the energy and ionize. And so we showed that we could detect it in a mouse infection. Miyang Nunt, one of her first papers, demonstrated that in microscopy negative pregnant patients that it detected more than half of them with this technique in the bloodstream, and that we could pick up all of the malaria species. So all we had to do is get blood, dilute it in water, and instead of having our eyes reading a glass slide, this was a laser reading on a metal slide malaria parasites. Although the machine to run it is tens of thousands of dollars and had to be reformatted. So that sort of sat on the table for a while. And this is an EM after low amounts of radioactive chloroquine that sort of shows 3 plumes right on the crystals, as if the drug is binding to it rather than being toxic all by itself. And so we showed that it was a saturable process binding to the crystals, but dependent upon heme. That drug by itself didn't bind to the crystals very well, but you sort of needed heme and drug to go ahead and bind. And in the late 2000s, Curtis Chong had spent about 6 months in Mozambique, and he was an MD PhD student, really wanted to work on malaria by itself. And like many of my summer students, I started him on a heme crystallization assay. And he used it a lot, and so much that my spectrophotometer, the light bulb broke. And this was like 2 weeks before his presentation. So then he adapted the assay to a plate reader, high throughput, and was able to run lots of conditions and found out, well, one, he generated 2 notebooks about this much for his rotation, got a paper out of it, and, but showed some interesting reversible kinetics of heme crystal inhibition, in that it was time dependent and it was a reversible process rather than being irreversible. And that there's also a pH dependence to the heme crystallization. So before, this would have taken me weeks to generate the data, but with the new plate reader assay, we're able to get lots of this data fairly quickly. So in summary, right now we sort of have this process of making the heme crystal requires a pH less than 6. It can occur in a test tube with water, but in the parasite, we think it goes on in a lipid environment to exclude water so you can make the crystals. And you sort of make a head-to-tail dimer, then you form this larger crystal. And that inhibition is reversible with the quinolines, requires binding to heme, is pH dependent, and could either be that you're not making the dimer to go in or that you're binding at extra sites. So working with a crystallographer in Texas, sort of came up with this proposal on a kink site on a growing crystal. And so we sort of think that a heme drug complex binds into this growing site in red and sort of caps off the extension of the hemozone crystals. And fundamentally, this is how the quinolines, which bind heme, work to kill the malaria parasite. And that's part of the iron problem. You just upset this a little bit, and you can make the best parasite in the world, which is a dead parasite. In contrast to the quinolines, which stick around for weeks with effective parasite kill, the artemisinins only last for about 6 hours, and A is a short plasma exposure with the artemisinins. And we also know for to treat malaria, we're going after a short 3-day duration. So if you're treating tuberculosis, you're treating for 6 to 9 months. The half-life is 24 hours, so we sort of have this duration of drug exposure to doubling time of 168, triple figures. P-falciparum with the artemisinin, though, we are in single digits, barely 1. The quinine component, this ratio of duration of effective drug concentration to doubling time is in double digits in P-falciparum. In contrast to the short turnaround time of E. coli, we're in the hundreds for this duration of drug exposure over, so it takes a long time, a long ratio to effectively eradicate pathogenic organisms from the human body when they're causing damage by itself. So we had gone down to 3 days of the artemisinins, mainly because it was 90% effective. 5 days didn't add that much. 2 days of drugs was only 60% effective. So we sort of settled in on 3 days of drugs by itself, although the 3 days is a bit of a misnomer because we sort of count day 1, and then day 2 is 24 hours later, and then the third day up at the top is within this, in the blue line, 48 hours. So all 3 doses of our artemisinin drugs are under a single 48-hour life cycle. So it's like trying to treat tuberculosis in 1 day. It's like trying to treat E. coli in 20 minutes. It just doesn't work with a microbe, but we've been getting away with it with malaria therapy and the artemisinins. All drugs are dose and time dependent, and I sort of want to end, maybe the Brits had it right 200 years ago when they began to mix the malaria drugs with sugar and gin. It was much more concentrated back then, the quinine, and I guess it was easier to go for a longer duration.